Thank you very much for the invitation. I was supposed to be in presence, but unfortunately, I didn't get my visa, so I'll have to give the talk online. So I'll be talking about our recent work on explaining conformational diversity in protein families using supervised and unsupervised learning approaches. This is a work performed by our PhD student from University of Sorbonne, Valentin Lombard, and co-supervised with Elodie Lane. Why are we interested in these questions? Because the classical protein structure prediction problem has been already solved, but proteins are not rigid, they are flexible, they interact with the partners, and they perform their function by changing their conformation. The flexibility, the conformational heterogeneity can be solved with a range of different techniques as molecular dynamics or normal modes or principal component analysis that will use just a few additional collective coordinates. And in principle, we can use this idea and apply it on the whole scale of the protein data bank. Just to give you a connection between molecular dynamics, NMA, and PCA, I will present two theoretical slides. So you've just heard about the molecular dynamics that solves the Newton's equations of motions. But these equations can also be solved slightly differently if we're not very far from the equilibrium position. So here we may expand the potential energy into the Taylor series. And if we're not too far, we can describe the potential energy using the second order approximation, which is the Hessian matrix. It turns out that if we substitute this expansion back to the Newton's equations of motions, these equations can be solved analytically in the closed form using the diagonalization technique. I can give you probably an illustration. So this diagonalization is rather similar to the latent variable space learned by the supervised learning approaches. But in case of the normal mode analysis, the latent space is obtained using non-supervised learning. And also very often when we mention the normal mode analysis, we typically also mean the elastic network model. In principle, all types of potential functions, all types of force field can be plugged in, but currently we're mostly using the elastic network model because it's very fast, it's very efficient, and there are very few or even zero adjustable parameters. The only adjustable parameters which is very often used is just the cutoff distance within which the atoms or the residues or the bits are connected together. Okay, to give you an example of the normal modes, typical motions will look like this, and these motions are computed in real time within 1 second on a personal laptop. So the computations are very, very efficient. And finally, my second theoretical slide about the connections between the PCA and the NMA. So the nice theory allows to converge from the normal mode space into the Cartesian space using the harmonic approximation. We can compute the mean fluctuations. Because of the harmonic expansions, many terms will cancel out. And very briefly, we can also compute the covariance matrix. The average between different x together, and if we expand the right side hand a bit, so L here are the normal mode vectors and omega are the frequencies, we can see that the right hand side can be expressed as the inverse of the Hessian matrix. So basically, the result that we need to remember is that the positional covariance matrix equals or proportional to the inverse of the Hessian, or the Hessian is proportional to the inverse of the covariance, And we can also compute the normal modes from the positional covariance matrix, because the eigenvectors of both matrices are the same. Okay, so this is the only theoretical result we need to know. And it brings us to our work, which is called DANCE, the DANCE pipeline. The whole approach is based on the analysis of the protein data bank conformations. So we have analyzed about 700,000 of protein chains. If possible, we didn't use the PDB chains. We used those from PDB redo. We took all the experimental structures. We took the X-ray, we took the enema, we took the cryo-EM. For the enema, we only used the first models. We've clustered the sequences at different multiple identity levels, and then we aligned the sequences and also aligned the structures. After the structural alignment, we interpret the structural heterogeneity using the motion extraction, which I will present later. And this allows us to assess some classical manifold learning and also supervised learning techniques. The whole pipeline takes about 2 hours on a personal desktop. It's very fast. You can repeat it on your own machine, or you can download the pre-computed ensembles. And we have also repeated the pipeline for multiple combinations of sequence identity and coverage, starting from the very stringent setup of 80% of identity and 80% of the coverage to a very relaxed setup with 30% of sequence identity and 50% of the coverage. In most of the cases, in 50%, going from the stringent to the relaxed setup does not change the size of the conformational collections. But in about 5% of the cases, if we relax the setup, then the conformational collections increase by about 100 members or even more. Just to show you some examples of the conformational collections. So for the adenylate kinase, it looks on the right. And our idea was to interpret the conformational heterogeneity using positional covariance and continuous linear motions. These motions are computed using the covariance matrix. They can be represented using the arrows as shown in the middle, or we can also show them as a trajectory. So once we have extracted the linear motions, we estimated their complexity using the number of components which are sufficient to explain 90% of the positional variance. We have assessed the complexity on multiple levels of the sequence identity and sequence coverage. You can see results here, and we may conclude that overall, only 1 or 2 linear motions are sufficient to explain a half of the conformational diversity, which brings us to the first conclusion that experimentally resolved protein conformations lie on the low-dimensional manifolds, which allows us, in principle, to use classical manifold learning techniques to reconstruct these manifolds. Just 2 examples. So the ADK kinase has 1-dimensional motions with the first principal component, which explains 97% of the conformational variability, the conformational variance. And another example, the ATPase has a 3-dimensional motion because only 3 components are needed to explain 90% of the conformational variance. So another another observation is that the motion complexity does not really change over the identity and the coverage of the conformational collections. If we relax the sequence identity, we will have more conformations. So in the first examples, 28 more conformations in our collections, and the motion complexity increases by 5 principal components. However, it may happen that new large amplitude motions may lead to apparent simplifications of the variability. Here in the second example, we added 54 new conformations, but it has reduced the number of principal components by the 22. So overall, we don't see a difference in the motion complexity over the whole range of sequence and coverage. Now, the second question is, since our conformations lie on a low-dimensional manifold, can we use classical manifold learning techniques to learn them? To answer this question, we have constructed a benchmark which consists of 10 representative protein families with different levels of motions, starting from very simple motions, one-dimensional, so adenylate kinase, going to more complex motions with 3 modes, to even more complex motions, for example, as in homologs, which requires 14 principal components to explain 95% of the variance, to even more complex motions with 40 modes to explain 95% of the variance. The question we'd like to answer is, can we predict unseen conformations using classical manifold learning techniques? These techniques include the linear PCA, the nonlinear PCA, or even maybe some more recent techniques as UMAP, ISOMAP, or t-SNE. Unfortunately, we were not able to use ISOMAP and t-SNE because we cannot reconstruct original space from the latent space. We could only use PCA, kernel PCA, and UMAP. So what did we do? We clustered our ensembles. We used some of the clusters as the training set and other clusters as the test set. And we have measured the quality and the complexity of the reconstruction as the distance to the training set. We have here, we compare the linear and the nonlinear, the kernel PCAs, and we can see that overall we can achieve a very high level of the reconstruction, except a few families. So ITPs and ABCs are very large proteins, so they are harder to reconstruct. And calmodulin has very large transitions of 2 domains. It's also rather hard to reconstruct. Also, we have measured the quality of the reconstruction as the function of the distance to the training set. So at the distance to the training set, we use the distance to the convex hull of the training set. And we can make 2 conclusions here. So first of all, only 14 out of about 5,000 conformations lie inside the training set. Nearly all of them are outside, so we're always in the extrapolation regime. And secondly, the quality of the reconstruction is proportional to the distance to the training set. So if we're close enough, the reconstruction is of a very high quality. So very often we're below 2 RMSD angstroms of the reconstruction. But if we go further from the training set, the reconstruction quality drops. So the quality only depends on the distance to the training set. We have also assessed the stereochemical quality of the reconstruction. So we have tried different metrics. Here I show the Ramachandran statistics. So what we can observe here that for all the tested cases, UMAP is significantly worse than all other techniques. And otherwise, we don't see a big difference between the linear PCA and the kernel PCA. Sometimes linear PCA is better, sometimes kernel PCA is better. And very interestingly, sometimes the reconstruction improves the crystallographic quality because of the denoising effect. And finally, we also assessed the biological relevance of the learned representations. So for this, we've chosen the ATPase protein, and we interpolated a path in the latent space from one conformation to another conformations, and we've measured the distance to the known experimental intermediates. In blue, you see the kernel PCA. In green, you see the linear PCA. So you may see that for one intermediate stage, the kernel PCA achieves a better result. For the other intermediate stage, the linear PCA achieves a better result. And visually, these transitions look like it's shown on the left. For the linear PCA, the transitions are linear from the starting to the final state. And for the kernel PCA, we have some curved motions. We have also compared these curved motions to the nonlinear normal mode analysis developed in our team, so which is called NORP. And you may see that the kernel PCA obtains transitions which are visually very similar to those obtained by the NORP method. And now it's time to present you the supervised approach. We're moving to the third part of my talk, where our motivation was to use sequences as the input, predicting the motions as the output in a supervised learning regime. So our main motivation was the rise of the protein language models. What are the protein language models? These are unsupervised or self-supervised models that train some latent spaces or embeddings by solving a masking task. What does it mean? It means that we start from a protein sequence, we mask nucleotides. Here I show only one masked nucleotide. Then we construct a protein language model with an encoder and decoder architecture typically, with the task to predict what has been masked. And these models, they construct some latent or embedding or representation spaces, which can be used in some other downstream tasks. What I show here is one of the possible tasks of classifying protein sequences, but we may imagine many more tasks. For example, we're using protein language models to predict protein motions. In our model called SeaMoon, we start with a protein sequence. We use a pre-trained protein language model. We have tested several of them. We have tested the ESM2, we have tested the PROS-T5 and ESM3. I will detail about them later on. Then we construct a neural network using the pre-trained embeddings. Again, we have tested multiple architectures and we have chosen the one based on convolutions with about 1 million of trainable parameters. And we predict the linear protein motions. Later on, very recently, we have also extended the model and use the three-dimensional structure as an additional input. So I will also show some results of this one. So what do we predict? We predict conformational heterogeneity expressed as linear motions applied to each of the residues as a ground truth, extracted from the DANCE method, which I presented before. And we compare the ground truth to the predictions using the loss function that combines rotation, permutation, and scaling of the predictions. Why do we need rotations, permutations, and the scaling? I will try to explain it a bit better, because when we predict the motion, the SeaMoon model is a model working only with sequences as input. So it doesn't know what is the structure. It predicts the deformation vectors applies to each self-atom or to each residue and not the structure. So here on the left, we show the structure just for the illustration purpose. In reality, our prediction, well, prediction is shown in blue and the ground truth is shown in red. They are oriented in a global random frame. To train the model, we have to align this frame to the ground truth. And for the alignment, we are solving the best alignment problem in a closed form, which is also called the Procrustes problem, to find the rotation which aligns the predictions with the ground truth. So in our laws, we also relax it with the permutation and the scaling matrices because we do not actually care about the direction and the amount of the deformation. So both predictions in blue and in the pale green are equivalent. And we relax our loss functions. For this situation, we also add the permutation relaxation because we do not care about the order of the principal components. What is important to us is only the global linear subspace. Now, yes, I have to also mention that we normalize the predictions by the maximum possible value, and we have chosen a threshold of 0.6 as that for the correct predictions. So here the ground truth is in yellow, the predictions are in blue. So all of them look alike with the normalized loss shown on top. And we consider these predictions as acceptable. The normalized losses which are greater than 0.6, we consider them as incorrect. And now we go to the results. We have assessed 3 types of language models. So one is SM2, which is only sequence-based, and 2 others use implicit structural knowledge. The ESM3 is a multimodal model, which solves multiple tasks during the training time, and the PROST5 is a language model that also uses structural tokens. So both ESM3 and PROST5 has a certain knowledge, certain information about the 3-dimensional structure of the proteins. What we can see that on average, ESM3 and PROST5, the models that use structural information, perform better than purely sequence-based model. Again, I show the correct and incorrect prediction on the right, and I can also show you the normal mode analysis prediction for this protein, which is incorrect. We can also see that our model generalizes well over low structural and sequence identity. For example, we can get acceptable predictions, so those below the 0.6 threshold. For the test cases which have low structural identity, so TM scores below, for example, 0.6, and low sequence identity. Also, we may compare our predictions with the prediction from the normal mode analysis, and the normal mode analysis is the worst among the 4 compared models. We can also see that in principle, the normal mode analysis is complementary to the sequence-only SM2 model. So test cases that predicted well by ESM2 are not always predicted well by the NMA. Only 50% can be predicted by the NMA, and vice versa. If we predict well something with the NMA, then ESM2 does not predict them well. However, nearly 75% of the cases predicted by the NMA can also be predicted by the PROS-T5. And these are the cases which cannot be predicted. These are two cases which cannot be predicted well by the normal mode analysis. So in one case, this is open closing transition of the bacterial toxin. In the second case is a complex unfolding motion, which cannot be assessed by structure-based models like the normal mode analysis. And finally, we have also added some additional structural information as the input. We constructed a new model, PTMO. And in this case, providing additional structural information increases the performance of our model by about 10%. So our model is always better than the previous one, and we are much better than the normal mode analysis. So this is time to conclude. So we have analyzed all experimentally solved protein conformations and demonstrated that they lie on a low-dimensional manifold, which allows us to use classical unsupervised and supervised learning techniques. We've assessed the classical manifold learning techniques and demonstrated that they can predict dancing conformations, but only those which are in the close vicinity of the training set. And we have also constructed multiple models which are based on sequence-based embeddings and demonstrated that the sequence-based and the physics-based approaches are complementary and can predict structural heterogeneity observed in the protein data bank. So thank you very much. And this was again a joint work with the team of Sorbonne by Valentin Lombard and co-supervised by Elodie Len. Thank you very much.
Thanks for the talk, Sergei. Have we got any questions in the room? No, can't see any hands up anywhere. I'll just have a check on Slack. Yeah, we've got a question on Slack. I'll read that out for you. It's from Robert Offner. Concerning a PAE matrix from AlphaFold with some blank squares corresponding to domain motions of the protein structure predicted, could your methods shown here be used for filling out these blank squares with directional vectors explaining the domain motions?
Yes, sir. Yes, yeah. I mean, it can be applied to any protein sequence. Yes. Yeah, we can try. Yes.
Still can't see anyone in the room. We've got another question on Slack from Hesna Kara. Is there any size limit to predict the motion from sequence?
It's a good question. Typically, we see that the predictions are worse for bigger, larger sequences. In principle, there are no technical limitations, but the accuracy drops down when the sequence is longer than 1,000 of nucleotides.
Okay, thanks very much. Oh, we got one more question on Slack. We'll make this the last one. Is ligand-based conformational change predicted?
It's a very good question. Yes, it is predicted because this is what we have been using as the training. But what we haven't done and which has to be done is to separately assess ligand bound and ligand unbound cases. So we have never separated the two. We have used both bound and unbound cases as the training set. They are predicted. They are predicted.
Okay, thanks very much, Sergei. If we can
Thank you very much.